Hi everyone, it's Professor Connor. Welcome to week 4. It's crazy to think that we're already in the 4th week of the semester. So before we start doing anything, thank you so much for your hard work. Thank you for being patient as we work through all the little glitches and kinks that have come up as we're trying to do this online thing. It is not my first choice. It is so hard to teach a lab online, as you can imagine, but it's also hard as a teacher that enjoys interaction with their students to be in this environment. So thank you for being patient. Today I'm just going to show you what the module looks like and what you're expected to do so that you have a good, clear picture of the expectations for this week. But first I want to say thank you again for working super hard. All right, so I'm going to share my screen. Just quickly, I'm going to show Canvas. So here is week 4. We're doing biologically important molecules, which would include things like proteins and sugars and starches and carbs. So your lab will run from June 8th, which is today, until the 12th, which is Friday, at midnight. This week we do have McGraw-Hill labs. There are 3 of them. As you can see, they're highlighted here for you. This is your question and answer piece where you can go in and kind of bond with other people in the lab. These are the objectives that you're going to be meeting this week and what you're doing, your assignments. As always, a post-lab. This is the lecture, the PowerPoint that I'm going to be going over. There's the solid point of the PowerPoint. And then this is the transcript for that PowerPoint, and of course the quiz at the end. So for now, I'm just going to start on the PowerPoint. So I'm going to start here. Okay, so like I said, this week we're doing the biologically important molecules, exercise 6 in your lab book. For this week, we're learning about the structure of biologically important molecules and how we test for them in unknown solutions. Biologically important molecules refer to the molecules that we make up the main structures and carry out primary functions within living systems. And these include carbohydrates, proteins, lipids, and nucleic acids. As we go through, pay special attention to the molecular structure of each, how they are constructed, and how each test reacts with the molecule it is testing for. Also practice identifying the positive and negative controls of each test. Although you won't be doing a virtual lab for lipids or nucleic acids, you will still need to know the structures and the tests that go with that. So before we talk about our molecules, let's review a couple of important chemical reactions and review positive and negative controls. The first is called dehydration synthesis, which is what we're looking at now, in which has small molecules are built into larger molecules through the removal of water. Dehydration means removal of water, and synthesis means to combine. So during the reaction, a hydrogen ion from one molecule will bind to the hydroxide ion to form a water molecule, and right where those ions are removed, a covalent bond forms between the 2 smaller molecules. So we're looking at this over here. The second reaction is hydrolysis. So during hydrolysis, a water molecule is added to break a larger molecule into smaller molecules. Hydro means water, lysis means to break. It is the opposite of dehydration synthesis. The hydrogen ion from the water will be added to one molecule, and the hydroxide ion from the water will be added to another molecule. And there we have it there and there. So controls are very important in an experimentation. They validate our results by ensuring our tests are working and that it's only reacting with the variables we're testing for. In this week's lab, we will use both positive and negative controls. The test tubes shown here contain the solutions listed on table 6.2 on page 58 of your lab manual. A positive control is a solution that we know before the experiment that it contains the variable that we are testing for. So it illustrates what a positive test result looks like so that we have something to compare it to when we're looking at our results. It also ensures that the test is reacting the way that we would expect it to. And negative control, in contrast, is a solution that we know does not contain the variable. Normally, it contains the solvent we used in our other solutions. And for this week, that would be water. It illustrates what a negative test result looks like so that we can compare it to our other test results. It also allows us to ensure the solvent doesn't react with our test reagent. Carbohydrates is the first thing we're going to talk about. Carbohydrates are sugar molecules used as an energy source for plants and animals. It's our fast energy. Plants store carbohydrates in the form of starch, and animals store it in the form of glycogen. It's composed of carbon backbone bonded to hydrogen and oxygen, and it forms a ring structure, as we see here. A simple sugar monomer is called a monosaccharide, but it can form disaccharides and polysaccharides through dehydration synthesis, which means they're just a monosaccharide that's built upon each other. In this week's virtual lab, we do 2 tests for carbohydrates that each test for a different kind of carbohydrate. The first is the Benedict's test, which tests for simple sugars. And the second is the iodine test, which tests for starch. So in other words, simple sugars that are broken down is the Benedict's, it has to be broken down. And starch is the iodine test where it is several monomers basically clustered together. It hasn't really broken down yet. Some simple sugars, like glucose and fructose, are what we call reducing sugars. This means that they have the ability to reduce weak oxidizing agents like copper. So the Benedict's reagent we use for the Benedict's test contains cupric acid, or cupric ion, so it's a Cu2+, which becomes red or reduced to cuprous, which is not just Cu+. In the presence of the reducing sugar and heat. So note that starch is not a reducing sugar. This results in the color change from a blue to either a green, orange, or red, depending on the concentration of the reducing sugar. This is also discussed in your lab manual and virtual lab, or in the virtual piece that we have in the lab for this week. The test results shown here are from procedure 6.1 in your lab manual and contain the solutions listed in 6.1 plus the Benedict's reagent. In this example, we can see that in tubes 1, 2, and 3, and 4, and 6 all contain varying concentrations of reducing sugars, while 5 doesn't. It has nothing in it. Amino acids, the second test for carbohydrates is the iodine test, which reacts specifically with starch. Starch is a very large polysaccharide made up of glucose monomers. This long chain forms coils that are the basis for its reaction with iodine. So when the iodine interacts with those coils, it produces a color change from a yellow to a purple and a black. The example shown here in procedure 6.2 and 6.1 contain solutions listed in that 6.1 plus iodine solution. In this example, we see that spots 2 and 7 both contain starch and the others didn't. So you have a variation of this color, that darker color, and this really has a lot of starch. Amino acids are linked together by polypeptide bonds to form proteins, sometimes called polypeptides for that reason. So see figure 6.7 in your manual for the dehydration synthesis reaction. Amino acids are composed of a central carbon atom bonded to 4 side chains, an amino group, a carboxyl group, a hydrogen, and a variable side chain indicated with the R right here. That is actually the amino acid. The test we do for proteins is called the Barut test. It reacts specifically with the peptide bond formed between the amino acids, which means it reacts with individual amino acids. During the Barut test, a cupric ion present in the Barut reagent will complex with the peptide bonds found in the protein and change to a blue or a violet. This example comes from procedure 6.3 in your lab book. The test tubes contain the solutions listed in table 6.2. So in this example, we see that both test tubes 1 and 5 contain protein, whereas 4, 3, and 2 do not. Tubes 2, yellow color comes from a honey solution and is not related to the test itself. Lipids are more variable in structure and function compared to proteins and carbs. The structures most relevant to our class are triglycerides or fat molecules, and phospholipids, the molecule that our cell membrane is made out of. A triglyceride is made out of one glycerol and 3 fatty acid bonded by ester linkages, and phospholipids are made out of 1 glycerol, 2 fatty acids, and a phosphate group. Although you won't be doing tests for lipids in your virtual lab, there are 2 tests for them that are important for you to understand. They are the solubility test and the Sudan 4 test. The solubility test is based on the fact that lipids are hydrophobic, so nonpolar molecules. Because of this, they're insoluble in polar solvents such as water, but they are soluble in non-polar solvents such as acetone. In this example, you can see that there is a small layer of oil floating on the top of the tube on tube 1. In test tube 2, the lipid has dissolved because of the acetone. So the Sudan 4 test is also based on lipids nonpolar characteristics. So in this test, Sudan 4 stains lipids red and then floats on top of the solution. The test is used in food labs when testing different foods for the presence of fats. The test results shown are from procedure 6.5 in your lab manual and contain the solutions listed in table 6.3. So we can see that tubes 5 and 2 had positive test results. Table 6.3 tells us that the tube 5 had a known lipid solution, which means that tube 2, salad oil, also contained lipids. Tube 3, which contains honey, had very little lipids compared to the salad oil in tube 2. So lastly, we're looking at nucleic acids. And nucleic acids are DNA and RNA where gene shortage and expressions take place, or gene storage, not shortage, sorry. The structure you see here is one nucleotide in an RNA molecule. Each nucleotide is made of a pentose sugar, which is either deoxyribose for DNA or just ribose for RNA, a nitrogen base, and a phosphate group. The test done for a nucleic acid is called a DDD test. The specific test for DNA, it's a specifically tested for DNA and will change from a clear to a blue if DNA is present. In today's lecture, we've talked about chemical reactions that build and break down large molecules. We also reviewed how to identify positive and negative controls in an experiment, and then went into detail on the structure and tests for carbs, proteins, lipids, and nucleic acids. Be sure to review the information in your lab manual and virtual labs for the week, and always feel free to reach out to me if you need help. This is kind of a long lecture. It goes through a lot of information. I would definitely look through those and memorize and be able to tell which tests go with which. So what test are we using to decide whether or not a carbohydrate is in something, or if a lipid is there? So those are all big pieces to look at. And then making sure I know what a positive control is versus a negative control. So I'm just gonna exit out of my lovely PowerPoint. So big pieces today. Know what each test does and what colors are what they are supposed to look like. What test for what? What test do we use again? Do we use for proteins? What test do we use for lipids? Also, just make sure you, like I said, have those definitions down. What's a positive control? Positive control gives us the correct reaction so we know and can compare our results to that positive control. Negative control shows nothing. It shows that there's nothing in that sample. So just keep those things in mind. As always, make sure you're reading through everything. If you have questions, contact me. I'm here. If the office hours do not meet with your schedule, then you can always do a Zoom with me. It's simple, easy, and it can be anytime. So please let me know if you need help. Please don't stay in the dark on these topics. This is actually, like I said, a pretty straightforward lab, and hopefully it will go well this week, better than others. Again, if you need help, let me know, and keep doing such a great job. You guys are amazing. I love you all, and you're going to do great this week. So have a great week. See you on Friday when we're done.